Hello and welcome to Unacademy, a one-stop destination for all English medium UPSC aspirants. As a part of NCERT fundamentals, today we will be discussing a very interesting and a very important topic, microorganisms. The chapter 2 from 8th class Science NCERT. But before we proceed with the session, a very important announcement. We have 3 comprehensive batches for the year 2025. The English medium batch Achievers, the bilingual batch Summit and the Hindi medium batch Shikara. All of them starting on 26th July. And a long-term NEEV batch for the year 2026 starting on 27th July. Most importantly, all these courses are available at just 23,999 for 15 month subscription. Do enroll before the batches start. Let us now continue with our topic, microorganisms. Firstly, what are the microorganisms? Microorganisms are those that cannot be seen with the naked eye and can be seen only with the help of a microscope. They are so small that we cannot see them without a microscope. And how important the microorganisms are? To estimate the significance of microorganisms, let us see a small point. During the initial days of evolution, the microorganisms lived on this earth without us because we were not yet evolved by that time. But we cannot survive without microorganisms. This explains how important the microorganisms are. Let us now discuss about the types of microorganisms. Microorganisms can be broadly divided into 2 types. Firstly, single cellular, also called as unicellular. Uni stands for 1. And the second type is multicellular. Multi stands for many. Their body is made up of many cells but not a single cell. And the unicellular microorganisms made up of a single cell. The single cellular organisms can be further divided into 2 types. Prokaryotic. They are unicellular. Their body is made up of a single cell, but that one single cell do not have a true nucleus. Therefore, they are prokaryotic microbes. Example, bacteria. And the second type is eukaryotic. They are also unicellular. Their body is made up of a single cell, but that one single cell will have a true nucleus, means eukaryotic microbes. Example, some algae and protozoa. Now, coming to the multicellular organisms. Their body is made up of a few cells, not just one cell. And examples for multicellular microbes are some algae and fungi. Then you may get a doubt. Algae are given as an example of multicellular microbes and also unicellular microbes. True. Some algae are unicellular and some algae are multicellular. These are the 2 types of microorganisms. In addition to these microbes, we should also see about the viruses. But viruses cannot be called as organisms. They are considered as a threshold between. Threshold means border. They are considered as the border between life and non-life. Why like that? Because when they are inside the host cell, means for example, if a polio virus infect a human being, then inside the human cell, that is called the host cell, inside the host cell, they are living. Fine. But how can we say that they are living? Because inside the host cell, they can replicate. And when they are outside the host cell, for example, when the polio virus is outside the human cell, they are non-living. Fine. But how can you say that they are non-living? Because they cannot replicate when they are outside a cell. And replication or reproduction is considered as the characteristic feature of a living organism. Anything that cannot replicate or reproduce cannot be called as a living organism. So the viruses are considered as the borderline between living organisms and non-living things. This is about the viruses, and these viruses are acellular. A prefix means absence, not unicellular, acellular. It means they do not have even a single cell also. The entire body of a virus is just made up of two components. One is a protein coat, a coat made up of protein, and then inside there will be some genetic material. So a virus is just a small piece of genetic material packaged inside a protein coat. This is the structure of a virus. The viruses can be divided into two types depending on the type of genetic material. The first type are called as RNA viruses. Right. What do you mean by an RNA virus? Inside this protein coat, if they have RNA as the genetic material, then they are called RNA viruses. Inside the protein coat, if they have RNA, ribonucleic acid as the genetic material, then they are called RNA viruses. These RNA viruses can be further classified into two types. The first type are called as retroviruses. Retroviruses. Listen carefully. Retroviruses contain RNA as genetic material. What is RNA? Ribonucleic acid. They have RNA as genetic material. What is genetic material? That material which decides all the biological features of an organism. For example, our genetic material decides our skin color, hair texture, height, health, all these aspects. So something, a material that decides the biological features of an organism is called as genetic material. These retroviruses contain ribonucleic acid, RNA as their genetic material, and they convert RNA into DNA and they inject their DNA. It means the virus will inject the viral DNA into the host cell DNA. For example, HIV viral DNA, HIV virus DNA will get injected into or added to, inserted into the human DNA. Let us suppose if a HIV virus infect the human being. HIV virus originally contain RNA genetic material, but after entering into human body, it will convert its RNA into DNA. Then the HIV virus DNA will get inserted, added to the human DNA. Such viruses are called retroviruses. One example is HIV virus and the second example is HTLV virus, human T lymphotropic virus. Okay. Now coming to the second type of RNA viruses. I remind you, RNA viruses are two types. One retrovirus, the second type is simple RNA viruses. Right? Retrovirus also contain RNA as genetic material. This RNA viruses also contain RNA as genetic material. Then what is the difference between these two? Both of them have RNA as their genetic material. Then what is the difference? RNA viruses retain the RNA. It means this RNA is not converted into This RNA is not converted into the DNA. Whereas in the retrovirus, the RNA gets converted into DNA and the DNA will get inserted into the host cell DNA. But here that will not happen. They have RNA as genetic material and they continue to have RNA as genetic material. They retain this RNA. Such viruses are called as simple RNA viruses. It means all RNA viruses are not retroviruses, but all retroviruses are RNA viruses because they all have RNA as genetic material. Okay, what are the examples of RNA viruses? It is difficult to give examples. Reason is most of the animal viruses, including the humans also, because human is also an animal. It means most of the human viruses are the RNA viruses. It means flu, coronaviruses, polio, Zika, Ebola, Nipah, all these are RNA viruses. Most of the animal viruses are RNA viruses. Now, coming to the second category. First category we have seen RNA viruses. The second category is DNA viruses. What do you mean by a DNA virus? Inside the protein coat, if you have DNA as genetic material, then they are called as DNA viruses. What are the examples for DNA viruses? Herpes, smallpox, that virus which is eradicated when Edward Jenner discovered the first vaccine. The first vaccine was made for smallpox. We do not have smallpox now. It is a disease which is already eradicated. And then hepatitis B, this is a liver infection. And adenovirus, this is a respiratory infection. And HPV means human papilloma virus. As you all know, the human papilloma virus causes cervical cancer. These are all the examples for DNA viruses. It means they have DNA as genetic material inside the protein coat. And retroviruses. As I already told you, HIV, the human immunodeficiency virus that cause AIDS, acquired immunodeficiency syndrome. Plus one more example is human T lymphotropic virus. These two are retroviruses. They have RNA but they convert their RNA into DNA. Next, remaining most of the human viruses are RNA viruses. This is all about the viruses. Now, let us discuss where do these microorganisms live. And these microorganisms can live in different places. They are everywhere. And some of the microorganisms can live in some extreme conditions also. Such microorganisms are called as extremophiles. Extremophiles. The word phile means love and extremophile means they love to live in the extreme conditions. For example, thermophile can live in extreme hot conditions. Psychrophiles live in very low temperature. Next, alkalophile, you know alkali means as a base and phile means love. They love to live in basic conditions, that is above the pH 9. And acidophile, they love to live in acidic conditions, that is at pH 2 to 3. And halophile, halogens are salts. They love to live in high salt conditions. And piezophiles, the word piezo means pressure. So piezophiles love to live in high pressure conditions. And metallophiles, metal, phile, love. They love to live in high metal concentrations. And radiophile, they love the radiations. I mean they can comfortably live under the high radiation levels. That is when UV rays are in excess or some X-rays are in excess. In that situations also they can survive. They are called as radiophile. And microaerophile. Examine the word microaerophile. Phile means love, micro means less, aero means air. Air in this context means oxygen. When the oxygen levels are less than 21%, these microorganisms can live. For all these microorganisms, a common word that we can give is extremophiles. Fine. Why are we discussing this? Is this information particularly given in the 8th class science NCERT? No. But microorganisms is given. So I thought of giving this extra information because there was a previous year question in the year 2023. The question is, read the question carefully. Consider the following statements. Some microorganisms can grow in the environments with temperatures above the boiling water. What are they? They are thermophiles. Is this correct? Yes. We have thermophiles. Microorganisms can live above the boiling point. Sometimes we just bring some food from outside and we heat it to some 60 to 70 degrees centigrade and we will be happy that all microbes are dead. Or we sometimes boil the water and we will feel very secure that all microbes are dead. But no. Beyond the boiling point of water, these microorganisms can stay alive up to 300 to 400 degrees Celsius also. They are called thermophiles. Next. Some organisms can grow in environments with temperature below freezing point. They are called as psychrophiles. Extreme cold temperatures. Yes, we have such organisms also. It means when we refrigerate also the food will spoil. Why? Because of this psychrophiles. They can live under cold conditions. Next, some microorganisms can grow in highly acidic environment below pH 3, acidophiles. Yes, we have acidophiles also. So this statement is also correct and the right answer is all the three. Such questions can be asked from the microorganisms, so I just thought of adding this particular information. And now, let us now discuss about some microorganisms and how they can be used. In the beginning of this class, we thought about how important the microorganisms are. Once again, let us recollect that point. We can only eat curd, but we cannot make the curd. But microorganisms can make the curd. What are those microorganisms? Lactobacillus bulgaricus, lactobacillus. The word lacto means milk. Bacillus means a rod-shaped bacterium, Lactobacillus bulgaricus. Streptococcus thermophilus. These are the bacteria. When you add these bacteria to the milk, they ferment the milk into the curd. That is how we get the curd. And coming to cheese, Lactococci. Again, lacto means milk, cocci means round shaped bacteria. Bacilli means rod shaped bacteria. Lactococci, lactobacilli, streptococci and propionic acid bacteria. These bacteria can convert the milk into cheese. So the raw material is the same, milk. But if you add these bacteria to it, it will become curd, and if you add these bacteria to it, then it will become cheese. Actually, they were accidentally produced. Like in some laboratory, a scientist forgot and left the milk like that. After some 1 week, 10 days, when he went back, he was surprised to find cheese, and he found the flavor to be pleasant, and the cheese found its commercial application today. And today we have varieties of cheese across the world. Next, bread. A yeast called as, not bacterium, yeast. Okay, and this is a eukaryotic microbe. Yeast is a eukaryotic single cellular microorganism. The name of that yeast is Saccharomyces cerevisiae. Saccharomyces cerevisiae. And how the bread is formed? When you mix the dough, in that when you add this yeast, that is Saccharomyces cerevisiae, then it will cause, the dough will have sugars, right? Like glucose, fructose, sugars will be there. Otherwise, we will also add some sugar to it from external sources. Okay. So, this sugar will be acted upon by this yeast, or the yeast act on this sugar, and it will ferment the sugar and will produce carbon dioxide and alcohol. This carbon dioxide is responsible for the air gaps in the bread. The more carbon dioxide it is released, the more fluffy the bread will become. And if you don't want to consume alcohol, then you should not consume bread also, because bread also contains a little bit of alcohol. The yeast Saccharomyces cerevisiae, which is responsible for raising the dough in the bread, is also producing some alcohol in that. That is the story of the bread. And some alcoholic beverages like wine. When a grape juice is added with some microorganisms, these microorganisms will ferment the fructose sugar in the grape juice and will convert this into wine. And these are again yeast. Look at this. The same yeast which we are using in the bread, the same yeast is used in the manufacture of alcoholic beverages as well. And then vinegar. We use vinegar for tasting purpose and also for preservative. As a preservative also we use vinegar. And this is chemically acetic acid. What is vinegar chemically? It is acetic acid. So, acetic acid bacteria, okay, will produce this vinegar. So, these are all different commercial products that can be produced by these microorganisms. And then, now, let us discuss about another concept called as normal flora. What is normal flora? Normal flora means some friendly bacteria. The friendly bacteria which we have in our body. Where in the body? Everywhere. Hands, skin, in the mouth, inside the digestive system, the small intestine, large intestine. So these microorganisms are present everywhere, but they are not pathogens. By the way, what do you mean by a pathogen? Pathos means disease. Gen means cause, something that causes the disease. They are pathogens. And these normal flora, they are not pathogens. They do not cause disease. They are our friends. Right. We have normal flora in our body, but in our daily excretion and other processes, we lose these friendly bacteria. So, it is very important to refill or replenish these friendly bacteria. For that reason, we have to consume probiotic food. Probiotic food. What is probiotic food? The food which is rich in friendly bacteria. The food which is rich in friendly bacteria. Okay, what are some examples of probiotic food? Curd is a very good example for probiotic food because it will have living bacteria in that. But these bacteria are our friends. What are they? Lactobacillus, bulgaricus, streptococcus, thermophilus. These are a part of our normal flora, the friendly bacteria. Same way lactococci, lactobacilli, streptococci, propionic acid bacteria. So, curd, cheese, these two are very good friendly bacteria sources. Therefore, we call them as probiotic food, the food which is rich in friendly bacteria. But bread, of course, we use the friendly bacteria in that, but when you bake the bread, these friendly bacteria are already killed. So that is the reason why we cannot consider the bread to be a probiotic food. So curd and cheese are very good probiotic foods, right? We also have another concept called as prebiotic food. What is prebiotic food? This food do not contain the friendly bacteria, do not contain friendly bacteria, but will support their growth, will support their growth. What are the example? Oats, fruits, vegetables, high fiber food, any food which is rich in the fiber. It will not contain the friendly bacteria, but it will support the growth of the friendly bacteria. Such food is called as prebiotic food. And now, what are the other applications of these bacteria? Where else they are used? These bacteria are used in cleaning up of environment. They are decomposers. You might have studied in ecology that we have three levels of organisms. Producers. By the way, we should not call plants as producers. Reason is, according to thermodynamics, energy can neither be created nor be destroyed. So the better word used for plants is transducers, because they convert radiant energy into chemical energy. Plants, they perform the photosynthesis. And then we have consumers, that is we all, okay. Herbivorous organisms, carnivorous organisms, we all are consumers. The third level is decomposers. So, you think these microorganisms comes under which level? They mostly come under decomposers category. Bacteria and other microbes, they are decomposers. Okay, all the three are compulsory for an ecosystem to run. All the three are important, but two components are compulsory, without which the ecosystem cannot survive. What are they? Producers and decomposers. It means plants and bacteria. It means even if consumers are absent, the ecosystem will not collapse. Of course, consumers are also important, but they are not as important as plants and decomposers. These two are compulsory element. The ecosystem cannot continue to exist if the bacteria are absent. Reason is cleaning up of environment. The dead and decaying organic matter, the animal feces, organic waste, all these materials should be decomposed by these microorganisms. And then we also use microorganisms in the preparation of medicines, antibiotics. Okay, let us discuss about antibiotics. Examples: penicillin, streptomycin, tetracycline, erythromycin, etc. And don't think that these are complicated terms, they are from the 8th class NCERT, please remember that. Okay, and now discussing about antibiotics. These antibiotics are produced by the antibiotics. They are produced by bacteria and they kill the bacteria. Yes, you heard me right. One bacteria will produce the antibiotics and they will kill another bacteria. A bacteria A produced this antibiotic, like say for example penicillin, etc., or streptomycin or something, and it will kill another bacteria called B. Why? Because competition, survival of the fittest, Darwin. Is it clear? And in between what we are doing, the selfish human beings, okay, we take up these antibiotics and use them as medicines. Use them as medicines. That is antibiotics. They have a property of killing the bacteria. So we take them as tablets to kill the bacteria that cause diseases in us. That is the story of antibiotics. Okay, and so, right. And not only the bacteria, some fungi also produce the antibiotics. In fact, penicillin is produced by a fungus called as Aspergillus, right? Okay, then Alexander Fleming discovered penicillin. Okay, the first antibiotic discovered was penicillin and Alexander Fleming discovered this. Okay, and we are discussing the applications of microorganisms. We have seen that they are the decomposers and an ecosystem cannot continue to exist without them. They clean up the environment. They are used in the medicines. They are used in the bread, curd, cheese, vinegar, alcoholic beverages. Is it clear? Now, next, biofuels. The next application of the microorganisms is biofuels. In the context of the pollution increasing and the depleting fossil fuels, alternative fuels are very important. In the year 2023, there was a mains question in the science and technology on the biofuels. It is that important. And also there were previous questions in the prelims about the biofuels. What are the biofuels? For example, bioethanol is a biofuel. Bioethanol is a biofuel. How to produce bioethanol? Fermentation. Who will produce bioethanol? Bacteria. So, these bacteria are used to produce the fuels. Okay. Next, bacteria can also be used in the manufacture of vaccines. What is the basic principle of vaccines? What is the basic principle of vaccines? Okay. For example, if you take polio virus, a vaccine for polio virus will contain a medicine for the polio virus or the polio virus itself. Yes, the vaccine will contain the virus itself or the vaccine will contain the pathogen itself. It means this is just like digging the well before you feel thirsty. It means producing antibodies, and don't confuse between antibiotics and antibodies. Antibiotics are the medicines we take, the tablets we take, like penicillin, streptomycin, amoxicillin, tetracycline, this kind of tablets that we purchase and consume. These are the chemicals. They are called antibiotics. Whereas antibodies are produced by our immune system. They are not the drugs that we consume. Okay. So, vaccines concept is about producing the antibodies before we get infection. Just like we have to dig the well before we feel thirsty, we cannot keep on digging it after you start feeling thirsty. Same way, you cannot start producing antibodies after you get infection. For example, recently we were discussing about this Chandipura virus. It killed 20 children in Gujarat 1 or 2 days back. What is the reason? That particular virus is killing. It will cause death within 24 hours. If you get infected by Chandipura virus now, within 24 hours the person will die. So, do we have time to produce antibodies after the virus enters into our body? No. So, we have to produce antibodies before we get the infection. So, what they do? For example, they inject a smaller dose of polio virus willingly. Killed virus, we take the virus, kill it and inject, we call them as killed vaccines. Or we take the virus, inactivate it and inject, we call it as inactivated virus. Or you take only one part of it, not the complete portion, only one part of it and inject. We call them as subunit vaccine. In this vaccination, we are cheating our body, we are misleading our body. The body will think that really the pathogen entered and innocently it will start producing the antibodies. These antibodies will be stored and if the virus really cause infection, these antibodies can go and kill those pathogens. This is the principle of vaccines. So, microorganisms are very important in vaccinations as well, right? Okay, so I remind you, we are discussing about the applications of the vaccine. The last application that we have seen is the vaccine manufacture. Now, let us try to understand the role of bacteria in the nitrogen cycle. What is the role of bacteria in the nitrogen cycle? Do we have abundant nitrogen in the atmosphere? Yes. 78% of our air is nitrogen, 20% is oxygen, and the remaining all gases are only in the minute concentrations. So most of the air is nitrogen. We have abundant, enough nitrogen in the atmosphere. But the problem is, this atmospheric nitrogen, or also called as molecular nitrogen, is in the form of N2, means N triple bond N. This triple bond is a very strong bond and cannot be broken easily. Cannot be broken easily. So, we have so much of nitrogen in the atmosphere, but it is useless. I mean, animals and plants cannot use this molecular nitrogen, that is N2, cannot be used by the plants and animals. Then, some microorganisms, they are called biological nitrogen fixers. What do you mean by nitrogen fixation? Look at the word nitrogen fixation. It is the process by which molecular nitrogen or atmospheric nitrogen, that is N2, gets converted into those nitrogen compounds, okay, that can be used. I repeat, nitrogen cannot be used by the plants and animals. So nitrogen fixation means converting the nitrogen into usable form. What is the usable form? Ammonia. So if you convert molecular nitrogen into ammonia, we call this as nitrogen fixation. Conversion of nitrogen N2 into ammonia is called nitrogen fixation. Who can do this? Not animals, not plants. Some microorganisms can do that. I already told you, microbes can do so many things that human beings cannot do. Microbes can do so many things that plants cannot do, right? These are called biological nitrogen fixers because they do nitrogen fixation. Now, let us see the nitrogen cycle and the steps in the nitrogen cycle. In the nitrogen cycle, the step number 1 is called nitrogen fixation. What is nitrogen fixation? I already told you, conversion of atmospheric nitrogen into ammonia is called nitrogen fixation. Okay. Who can convert this? Diazotrophic bacteria. Diazotrophic bacteria. What do you mean by diazotrophic bacteria? The bacteria that can fix the nitrogen. The bacteria that can fix the nitrogen. It means the bacteria that can convert N2 into NH3 are called as diazotrophic. Memorize, diazotrophic bacteria. Right. Okay. These bacteria, example, Azotobacter rhizobium. Azotobacter rhizobium. They can convert N2 into ammonia. They have a major role in this process. They are harbored in the roots of the leguminous plants. Leguminous plants means our pulses. These plants are called leguminous plants. Okay. In the roots of these plants as nodules. What are they called as? Nodules. These nodules will have Azotobacter rhizobium, the nitrogen fixing bacteria. So can a leguminous plant fix the nitrogen? No. The bacteria in the root nodules of the leguminous plant will fix the nitrogen, right? Then nitrogen fixation can also occur because of the atmospheric fixation, that is, through lightning. During the rain we get lightnings. That lightning also can convert N2 into NH3. Also we can convert N2 into NH3 in the industries, that is in the factory. Is it clear? So on the whole, N2 can be converted into NH3 by 3 ways. One is diazotrophic bacteria. Second one by lightning. Then we call it as atmospheric nitrogen fixation. This is biological nitrogen fixation. This is atmospheric nitrogen fixation. And we can also convert this nitrogen into ammonia in the factory or in the industry. So that is called as industrial nitrogen fixation. These are the 3 different ways in which you can convert nitrogen into ammonia. Okay. The second step in the nitrogen cycle is called ammonification. Ammonification. What do you mean by ammonification? Here also ammonia is released. In the nitrogen fixation also the end product is ammonia. In the ammonification also the end product is ammonia only. But what is the difference? Here it is not conversion of N2 into ammonia. Here it is, the plant and animal remains, dead bodies of the plants and animals, they are buried under the ground and they are full of nitrogen. How? For example, we have nitrogen in our proteins. We have nitrogen in our proteins because maybe you know the proteins are made up of amino acids. Amino means what? NH2. They have nitrogen. Is it clear? Amino acids, all the proteins, it means your hemoglobin, your insulin, your muscle protein collagen, your hair protein keratin, this is full of nitrogen. And our genetic material DNA and our RNA, ribonucleic acid, they also contain the nitrogen. Okay. So like this, living organisms fully loaded with nitrogen. So when a plant or animal die, when it is buried, all this nitrogenous substances, that is ammonia, chlorophyll, DNA, RNA, they will get converted into ammonia. That process is called as ammonification. Not nitrogen, not N2 into the nitrogen fixation is N2 into ammonia. This is release of the ammonia from the dead bodies. That is the difference. Okay. Now the third step is called assimilation. Whatever might be the sources, that is 1, 2, 3. Diazotrophic bacteria may release the ammonia, or lightning may produce ammonia, or it might be produced in the industry, or fourth, that is ammonia might be produced in the process of ammonification also. Ammonification. Okay, this will release ammonia, not converting, it will release ammonia from the dead bodies. These are the 4 sources of ammonia. Whatever might be the source, if that ammonia from all these sources is absorbed by the plant, plants will absorb it. If the plants absorb this ammonia, that is called assimilation, the process of absorption of the fixed nitrogen. Plant cannot absorb the molecular nitrogen N2. Plant will absorb the fixed nitrogen, that is ammonia, okay, through the roots. This process is known as assimilation. And then Finally, we have nitrification. What is nitrification? Plants assimilated the ammonia, right? Plant roots absorbed the ammonia. Then the plant will convert that ammonia into nitrite, NO2 minus. Okay, and who can convert NH3 into nitrite? Not the plant, again bacteria. Nitrosomonas, nitrococcus. What they do? They convert NH3 into NO2 minus. Okay, nitrosomonas and nitrococcus. Then next what happens is the NO2 will get converted into NO3 minus. That means nitrate. That is called nitrate. This is nitrite. This nitrite is not usable. Means plant cannot use it. But this is usable. That means plant can use it. Ammonia is also usable. I repeat. After assimilation, that is when the plant absorbed the ammonia, then nitrosomonas and nitrococcus will convert this into nitrite, and then nitrobacter will convert the nitrite into nitrate, which is a usable form by plant. So on the whole, plant will use NH3 and NO3 minus. These are the two substances that are used by the plant. Is it clear? And then animals will get them from the plants. Isn't it? When we eat those plants, we also get ammonia and nitrate. Plants will get it with the help of these bacteria, and we get ammonia and nitrate from the plants. After we all consumed, now the nitrogen should go back into the atmosphere. All this ammonia came from the atmosphere. This nitrogen should go back into the atmosphere to complete the cycle. That process is called as denitrification. Denitrification. Here, nitrate will get converted into nitrite, and then into the nitric oxide, then to the nitrous oxide, and then finally into the molecular nitrogen. It means this goes back into the atmosphere. With this, the nitrogen cycle is completed. Who will do this denitrification process? Again bacteria. Clostridium, Pseudomonas. Clostridium and Pseudomonas. They use nitrate instead of oxygen for the respiration. They are anaerobic bacteria. Anaerobic means they can live without the air. It means they can live without the oxygen. How? Okay, they use nitrate instead of the oxygen. What? Clostridium and Pseudomonas. Okay, so this is how finally the nitrogen enters back into the atmosphere. Now let us briefly summarize this point. First, nitrogen fixation, N2 into NH3, okay, either by bacteria or by industry or by lightning. That is called nitrogen fixation. Next, ammonification. Here what happens? Okay, not ammonia produced, but it is released from the decaying organic matter. Then next process is assimilation. Whatever might be the source, the ammonia is absorbed by the plants, and then the animals will consume these plants. They will also get this ammonia. Then nitrification, that ammonia gets converted into nitrate, which can be used by the plants and animals. Lastly, denitrification, nitrate gets converted into the molecular nitrogen. It goes back into the atmosphere, completing the nitrogen cycle. In every step of the nitrogen cycle, bacteria play a very important role. So it means bacteria are very important. Okay. Till now, we glorified the bacteria, but now let us see the harmful effects of the bacteria. Some bacteria will cause diseases. Disease causing microbes are called pathogens. Diseases caused by the pathogens are called communicable diseases because they spread through communication. It means they are infectious diseases. Infectious or communicable means they can spread from one individual to the other individual. Some pathogens spread through vectors, like say for example I mentioned about this Chandipura virus that we have seen recently, that will spread through sand fly and Aedes aegypti mosquito. So these are vector borne diseases. Same way dengue, chikungunya, malaria, they also spread through carriers. Female Anopheles mosquito carries the parasite for malaria. What is that? Plasmodium. What parasite causes malaria? Plasmodium. Is Plasmodium a bacterium? No, it is a protozoa. What is a protozoa? Eukaryotic microorganism. Plasmodium causes malaria and the Plasmodium is carried by female Anopheles mosquito. Same way, female Aedes mosquito acts as a carrier for dengue virus. These are vector-borne diseases. Next, we have microorganisms that grow in the food. See, once you prepare food and if you consume it after 2-3 days, you will get food poisoning. Why food poisoning occur? What spoiled that food in the 2-3 days time? Clostridium botulinum. Therefore, this kind of food poisoning is called as botulism. Botulism, because it is a food poisoning that occurs because of Clostridium botulinum, right? So like this, we get these diseases by consuming the food. Same way, tuberculosis, measles, chickenpox, polio, all these occur through air. They spread through air. Tuberculosis, measles, chickenpox, polio, they spread through air or air and water. Next, cholera typhoid, they spread through water. Hepatitis B spreads through water. Malaria spreads through mosquito. And what causes tuberculosis? Tuberculosis is caused by a bacterium. Measles, chickenpox, polio are caused by viruses. Cholera typhoid are caused by bacterium. Okay, Vibrio cholerae, Salmonella typhi, Mycobacterium tuberculosis. Hepatitis B caused by virus. Malaria caused by a protozoa called as Plasmodium. These are some harmful pathogens. Then, do these microorganisms cause infections only in the human beings? No, they will cause diseases in the plants also. For example, citrus canker is a disease for the citrus fruits, lime, lemon, etc. Okay, it spreads through bacteria and the plant will get this bacteria through air. Next one, rust of wheat. It is an infection to the wheat plant. This is caused by fungus. Fungi are multicellular microbes we discussed in the beginning. It will spread through air and also through seeds, contaminated seeds. Next, yellow vein mosaic bindi, okay, that is okra. It is also spread by a virus, okay, and it spreads through an insect which will, like pest we say, the pest that will attack this plant and that particular pest will carry this pathogen called as this virus, okay. And now lastly we are discussing about preservative techniques. We already understood that the bacteria are everywhere. So these bacteria are present in our food also. They can spoil our food. So how to preserve our food? One, add salt, but okay, that halophiles can still spoil the food. It means if you add more salt, for example pickles you take, they are fully loaded with salt, but still sometimes they spoil. Why? Because of the halophiles. Next, by adding sugars like jams and jellies. Vinegar, if you store some vegetables and all in the vinegar, that acidic pH, okay, vinegar's pH is less. So that will like kill the bacteria, but acidophiles will not die. So even if you keep the vegetables in the vinegar, they are not forever, they will still spoil because you have acidophiles. Oil, again fries, when you fry something, chips and all will not spoil so easily because oil is a preservative agent. Packaging, how packaging? Sometimes when you take a packaged food, okay, on that it is written that no preservatives are added. Then how it is preserved for 1 year? Milk, tetrafoil milk box. So in that you will have a shelf life of up to 3 months. 3 months you can use the milk. But on the package it is written as no preservatives. Then how can you store it? Simple. First boil the milk. Means kill all the bacteria which are existing in the milk. Then immediately pack it under the sterile conditions. So obviously when there are no bacteria inside that, bacteria cannot grow because new bacteria comes from the old bacteria. They are not created. So when you package this in the sterile conditions, that is also a method of preservative. Okay, fermentation. Okay, for example, grape juice will spoil but wine will not spoil. The older it is, the more valuable it is, isn't it? Hot and cold conditions, freeze. If you freeze the food or if you boil it, then also it is preservative. But we can also add some chemical preservatives. Sodium benzoate, sodium metasulfite are the commonly used chemical preservatives. And pasteurization. Pasteurization is a technique used in boiling the milk. It is called pasteurization because Louis Pasteur given this particular technique. What is the procedure here? Heat the milk above 70 degrees for just 30 seconds. Heat the milk above 70 degrees for just 30 seconds and then suddenly chill it. Suddenly chill it. Means what? You are exposing it to high temperature and immediately low temperature. Okay. So when you heat it to high temperature, okay, psychrophiles may survive but thermophiles may survive but psychrophiles will die. When you immediately cool it, okay, then the thermophiles will die. So it means this technique will kill most of the bacteria and our milk is stored by this procedure called as pasteurization. This is all I wanted to discuss about the microorganisms. Thank you so much.